our next speaker, it's my pleasure to introduce Dr. Paul Keown. Professor Keown is a specialist in immunology and transplantation and a physician at Vancouver General Hospital. He has served as the Executive Director for the BC Transplantation Program, President of the Canadian Transplant Society, and has head of the Division of Nephrology from 2004 to 2009. He has also been the director of the Vancouver General Hospital Immunology Lab for how long?
Long time.
Long time. Okay. Well, welcome, Paul, and we look forward to your talk.
Thanks very much.
Thank you. Thanks. So I'm going to keep my talk short because the morning is really exciting. We're having a lot of science, and we really want to expand Megan's time to hear all about the cutting edge of therapeutics. But what I would like to do is show you that the same kind of changes that we're talking about in molecular sciences are occurring in computational medicine, particularly in trials. So if we think about it simply, the world of molecular science is moving at rapid pace. We will never be able to test the advances using the old-style clinical trials. 2 arms, follow it for 2 years, stop, think about the analysis plan for the next one. Those days are dying and will be gone. And through the Genome Consortium, what we're hoping to do is to now take all of these innovations in computational medicine and build them in so we can do real-time analysis of innovations. So I'll show you some of the changes which are coming, the ones that we're actually building in now for our current genome, which is genome number 2, and then the ones that we hope to apply as we move forward to the next stage. So, on the left-hand side, we have the traditional approach to clinical research. We start with an area, let's say, transplantation. We ask, what are the interactions? What are the causal relationships? How do we determine causal consequence versus causal action? How do we look at treatment gaps? What do we think needs to be done? So perfect example this morning, antibody-mediated rejection. How do we treat it? What do we do? What will the future be? What's the understanding? All of these things. Then we come up with the answers, and now we want to move forward into clinical trials. And the classical trials are divided into 3 phases. Phase 1, which is very small groups looking for biology, looking for biometrics, looking for dosing, things like that. Then phase 2, slightly bigger group, maybe 100 or so. And then phase 3, usually the large trials powered for differences of a few percent. So we have 300 or 400 in each arm. They run into problems because it's difficult to get all the patients. They take a long time. They're enormously costly. I was just involved in the planning of a trial that's going to cost over $100 million to run. It's just phenomenal pricing. And then once we get the answers from those, now we have to introduce them into the clinic. So we present them to the FDA or Health Canada or whoever is the particular regulatory body of our choice at the time. They debate them for a while, and then they're not used in any formal approach. We all get the drug, we use it in different ways. So for us, it's even more complex because we're not talking about a drug. We're talking about a matching biology, and a matching biology coupled with alterations in immunosuppression to reduce toxicity, then the addition of new forms of cellular therapeutics on top of this. So the old style on the left will no longer work for us. We need to look at where the future is going. And if you only read one article in the field, I would recommend to you this one by Vivek Subbe. He's from ND Anderson. He's an oncologist. It's a wonderful article in Nature Medicine, and it really describes where the field is heading. So I'll show you some of the things. The top line here shows how things are typically done. We look, if we were working with drugs, we look for targets, we find a lead compound, we do some preclinical studies, we do the phase 1, 2, 3, then we take it to the FDA. That whole process, just the clinical process, typically took about 10 years to get through and literally cost hundreds of millions of dollars. The concept now is this little cluster of balls at the bottom which are moving around. which really condense everything so it can be done in real time. And the perfect examples are the COVID trials over the last 5 years, which took a time of up to 10 years that would normally take and condensed phase 1, 2, and 3 together, so that from start to presentation to the FDA, they were over in months rather than years, about 24 months rather than 5 to 10 years. Now, the concept of trials has changed entirely. In the past, it was we have a single question, drug A or intervention A, we have 2 arms, we follow them, we do the analysis at the end, we plan the next trial. Now they've moved into combinatorial trials, basket trials, bucket trials, where we either look at one drug in multiple diseases or multiple drugs in one disease, combinatorial treatments. And we can combine them in a whole variety of ways. But for us, we might say we have several things to look at once. We have epitope matching, we have epitope expression, we have a variety of other things. These are all used in a pool of patients so that we can begin to ask multiple questions and answer them all in parallel. There is a thing called patient centricity, which is very often ignored. It essentially says your patient is the most important part of your trial. And it's not something that's been really at the forefront of our mind. We pay lip service to it, but it's going to change because the consequences of failure for transplantation are high. So let's think where we're going next. Our next step is going to be the use of epitope matching to allow us to minimize immunosuppression. And all minimization trials to the present have resulted in failure and rejection, because in many people, we get the drug too low and the graft rejects. And once it rejects later, it's like waking of the dragon Smog in The Return of the King. It wakes up and it blasts everybody around it, and the graft dies. So we can't afford it. So now we have to have very strong understandings with our patient. We build things around. So let me address how I think it's going to evolve. This is one of the sort of premier COVID trials. The I-SPY COVID is really a group of about 30 centers around the world. It's led by Stanford and half a dozen other American centers, plus European centers. And they have brought together a large number of patients who flow in on the left side. And it says adults with COVID. And then they come and they either agree to be randomized to enter the trial or they don't. And if they don't, today they would be just let to go on their way home. But no, they're included in an observational cohort. So you say to the patient, will you enter this randomized trial? No. Will you enter an observational cohort? And most people say yes. So now you have the real world and you have the trial intervention. Once a patient comes into the trial, they're randomized into multiple arms, a backbone plus treatment A, plus treatment B, plus treatment C, and then the outcomes are measured. So at the same time, by using large numbers of patients drawn in from multiple centers, all in coordinated fashion, we can answer many questions. And this is where I think we would like to go with the Genome Consortium. We have now approximately 30 sites, exactly the same as the I-SPY Consortium, across Canada, the United States, Europe, and now Australia. We can begin to do the kinds of trials that in the past were completely impossible for us. And we can do them in a continuous fashion, so that where you see backbone, treatment A, treatment B, we can now continuously evaluate those and change the questions. But it's difficult even in a field where you can combine multiple sites. You're still limited by the numbers of patients that you can have. You want to use each one. Each patient is precious. And when we do a formal randomized trial, the most efficient mathematical model is equal randomization between two groups. It gives us the smallest total number of patients. So let's say we need 200 patients in each group for a sample for an effect size of X, so 400 patients. That means we are committing 200 of our precious patients to receive the same treatment that they would receive anyway today. It's not informative to us. But we can actually rebalance those, and we can reduce the number in the active arm if we have a way of using a comparator group which is reliable. So a lot of work now has gone into machine learning to try to develop simulation models. If we could simulate what this control arm would be like, we could then save those patients and put them into the active arm. So the question is, can we now build these models of the disease which are truly representative? And this is another example. Some of these come from Yale, some of them come from Stanford. They're really excellent groups. And many of them come from oncology because they have started this walk a little bit before us. So what they would do on the very left, where it says A, is emulate the trial. They would take all the patients they have, and they would ask, if we had randomized these patients into the trial and we expect this difference, what would the mechanics of the trial be? Then they divide their patients into synthesized treatment groups, compare them by continuous simulation. Then they emulate the way the trial would run, and they predict the survival analysis. So before they actually start the trial at all, they have a picture of what it would be like and the range of probabilities around those two survival curves. Now they want to say, okay, instead of having comparative patients running contemporaneously at the same time, can we actually use historical patients? Are they close enough to what we anticipate the future will be? Can we use those as our control group? To know that, you have to look at the changes over time. So we're very fortunate that very little has changed, let's say in BC, in the last 10 years. Our approaches to therapy have been standard, our approaches to patient selection have been standard, and we've actually done this, we've looked back over our patient data, and we can see that the interventions, the selection, and the outcomes of patients have really not changed perceptibly over that time. So we could now look back and use our historical data as a synthetic cohort to match against our new patients coming forward that we would enter. Some groups in the States have gone even further, and they've said, can we now generate a truly synthetic group of patients? Could we take some from here and some from McGill and some from Germany and others, pool them, and from those simulate a completely new population, which would be the synthetic population. Then we can all continue to add to them over time, and we continue to generate a truly synthetic group of control patients. So this is what we're planning for epitope matching. We want to be able to confirm that epitope matching either is beneficial or is not beneficial. We have retrospective studies that James addressed yesterday with large numbers, 1,000 patients, that show if we are rigorous in excluding sensitization, and if we look at those individuals who have close molecular matching at class 2, the risk of rejection is vanishingly low. It's approximating 0%. It's less than 1%, which to me is astounding because I mentioned yesterday, I come from the era when it was 80%. So now, given that, the question is, can we confirm that? So we want it to be safe and we want it to be sure, but we want to be fast because there may be different ways of matching. As Ruth mentioned yesterday, we can look at different degrees of matching. Should we match at DR? Should we match at DQ? Can we keep it simple? Does it have to be complex? So we want to ask a series of different ways of doing this. So the proposal is the following. We would look at a prospective group only, and for each patient we enter, we would add 4 retrospective synthetic patients who are matched to the new active patient. So the first one is Mrs. Jones. She's a 56-year-old lady with diabetes. She's been on dialysis for 3 years, been on the waiting list during that time. We look back into our now several thousand population database and we select 4 Mrs. Jones, and then they go in as her control counterparts. The next one is Mr. Smith. He's a 23-year-old man and so on. we find 4 Mr. Smiths. And you remember that I mentioned that the sample size might be about 400 for an effect size of 10%, 200 in each group. We can now actually begin to split those differently. So we can say we still need the 400, but now only 50, 60, 70, or 80 of them need to be prospective patients. And the other 300 or so can actually be the retrospective pooled synthetic patients. So we do 350 transplants a year here or more. That means we can do the first trial within the first 6 months very easily. And by having continuous data information, then we can begin to use automated stopping rules, which you see on the right. So we just let the trial run. We have the preformed statistical stopping rules. When we cross one barrier, it says, you have been successful, you can stop. And if we cross the other barrier, it says, this is worse, significantly worse than anticipated. In other words, riskier. Now you must stop because of treatment failure. So quickly we get treatment success or treatment failure. And as soon as we pass those barriers, we can begin to ask question 2. But we can go further because we can be doing this in multiple places at the same time. We can be doing it in Hanover, we can be doing it in McGill, we can be doing it in Toronto and other locations, so that we set up between us the questions we want to ask and we run the trials. So with continuous information input, continuous analysis, and automated stopping rules, we quickly come to an answer. Now, one of the problems we have is how will we actually share this data? And this is becoming more and more difficult. So the Europeans have moved to a very stringent code for privacy and data sharing, which the world is now adopting because otherwise we can't share data with our colleagues in Europe. But it has had such an effect that it takes a long time to get any approval for data sharing, and that is extremely stringent. In fact, in Canada, it's very hard for us to share data between provinces now. We can do it, but with great difficulty. We can do it if we centralize it, but if we want automated real-time data from the clinics itself, we don't want to centralize that data. So we have been working with our colleagues now you saw Clemens Buddha a few minutes ago with Clemens Buddha in the Charité in Berlin and several other groups, the Max Planck, and groups here across Canada to ask, can we do things differently? Instead of sharing data or pooling data, can we leave the data where it sits in each of our hospitals and share the algorithms for analysis? So through this NephroCage program, which has been very exciting, we've been using approaches which allow us to do that. So we've been using the same systems that are used for generating Bitcoin. I've tried to generate Bitcoin, it doesn't work, but we've used the same systems for generating Bitcoin so that we have clear security, clear separation of data and shared algorithms. And we're just coming to the point where we think this will work. So now we begin to think, what will the future be? We think that the next step in the Genome Consortium will be not just to transform science at the molecular level, but to transform testing at the clinical trials level. And it will be a radically new form of clinical trial with a radically new form of data analysis and integration that will enable us, we hope, to keep up with the speed of changes which are occurring. So I'll stop at that point, be happy to answer any questions for you.
Thank you very much, Paul. Do you have any questions? Yes.
Michael.
So this is fascinating work, how you moved the agenda for the field over the last 2 years, because you're so young.
You're so kind, you can come again.
Yeah. So how do surrogate endpoints, iBox, which is now on its path for regulatory approval, fit into these innovative trials? So how do you merge those, all 3 components? Better diagnostic tools and risk stratification, because the event, our classical events, get less and less, as you said.
Yes.
So there's one design around enrichment of risk populations where we can use biomarkers and others, but also to shorten the observational period through adequate surrogate endpoints. So how do we put this all together? And I think collaboration, of course, is a critical component. And that in the era of comprehensive and integrated medical records, which there were several papers in the US now that actually EPIC outperforms the STR registry in precision of getting demographic data about the US in transplantation. Right?
Yeah. So profound questions, Michael. Thanks. Maybe not everybody knows iBox, but it's a wonderful predictive algorithm that's been developed by the French group, now with input from multiple sites around the world. And the goal is to predict for each patient, according to baseline demographic and clinical characteristics, what their outcome will be. And this is really a sort of synthetic control in a way. It's just condensing the information from all of those synthetic controls into an algorithm that allows you to predict what the outcome should be for Mr. Smith. So if you like, it's the ultimate step in synthetic data. I have a little concern about it only because I think it's easy to predict population outcomes. So if you want to say, you know, Mr. Smith is a 40-year-old man who has these characteristics, what will the outcome for the group be? And yes, you'll be very close. But for Mr. Smith, if he stops taking his medications halfway through, the game is over. It's a different story. But these will become more and more important ways of amalgamating truly synthetic data. And then combining that with real-time informatics is going to make it very speedy to track these patients. So I think, Michael, just if I go one step further, I think Canada is a perfect country to implement these because the centers work well together. We operate in very similar ways. We have great ties and great collaboration. And I'm very hopeful that this will really serve as a model of what countries could do. I don't think it will appear in the States for a while because they're just a different animal. But Canada, I think, can really do this.
Right. In the interest of time and sticking to the agenda, let's stop here. And thank you very much again, Paul.